But I want to welcome you to the second event in our new Funding Fridays webinar series. And today, as you can see from this opening slide, is going to be an overview of the NIH R series application. And we're going to be holding these research and professional development webinars every month. Hopefully going forward, at least for this entire academic year. And it's usually on the first Friday of the month. There was a little bit of slippage for these first two webinars, but it's going to be on the first Friday of every month after this. And I also wanted to let you know that the Funding Friday webinar series, it's a product of the Pivot and Connect research initiative, that was launched just this past spring by our Associate Vice Chancellor for Research, Tamara Schneider. And this was directly in response to the COVID-19 health crisis, because the Office of Research was looking for ways to provide more targeted support for faculty during this difficult and unprecedented time. And we thought that this would be a good way of providing online training tools for faculty members. And it also represents a collaboration between a number of very valuable resources for grant seekers at our university. So I didn't introduce myself. My name is Effie McLaughlin. I am the Interim Assistant University Dean for Research, and I've been working in the Office of Research at Central Office for about 10 years now. And my portfolio includes many of the internal funding opportunities that our office offers, which many of you have probably applied for. But we're also collaborating for this webinar series. We're collaborating with the Research Foundation Office of Award Pre-Proposal Support, or maybe known to you as RFAPS, which is a very useful office in the Research Foundation, with the director John Sapogas and Dan Seatman, who are hopefully on this call. But they provide very valuable support in terms of not only grant writing, but they offer a peer review process. If you're working on a grant proposal, you can submit your proposal to them, and they will find people to take a look at your proposal before submitting it. And then also, finally, we are working with the CUNY Office of Library Services. And if you look at the entire program, the librarians that work with the CUNY Office of Library Services are presenting some important sort of professional development and grant prospecting and sort of research tools for you as well. So as I had mentioned, the goal of this webinar series is to create a living resource for research development and support and information for researchers across the university. And that's at every level, hopefully. Adjuncts, lecturers, junior faculty, senior faculty. And all of the webinars are going to be recorded. And actually, maybe Brandon, if you want to hop to the next slide for this, this is the consent. I did want to let you know that, all of the webinars are going to be recorded and made available on our Office of Research website. So if you're not comfortable with being recorded, please keep your camera off, keep your mute button off, don't provide a profile picture. Just so you know that it is. But the good thing about this is that you'll be able to come back and refer to this as you need it. We will make this presentation available on our website, also the recorded webinar available on our website. And we'd also, this is, as I said, it's intended to be sort of a living process. So if you have any thoughts or comments or suggestions about future webinars that you would like to see, please let me know, let us know. And even coming up with this, one of the discussions that we'd had is that the R series is obviously just one of the grant series that the NIH offers, and probably having a discussion about the SCORE grant program is also a worthwhile webinar for us to hold at some point. So if you're interested in that, please get in touch with us about the SCORE program. So allow me to briefly introduce our speakers. So Brandon Begarly is the Director of Research and Sponsored Programs at Lehman College, and he has a great deal of experience helping faculty members prepare NIH proposals. And he both has worked at CUNY for a long time in the Research Foundation and LaGuardia, and now at Lehman. And then also, before coming back to Lehman, he was at Vanderbilt University Medical Center, where his portfolio included working directly with faculty on NIH proposals. So he's very knowledgeable in this area. And then with him is Tom Kurtzman, who is an associate professor of chemistry at Lehman College. And he is very research productive, very research active. He was recently awarded an R01 from the NIH. And Dr. Kurtzman's role is primarily to complement Brandon's presentation by providing sort of insights about the NIH application process from a PI's perspective. So if you have, so he'll probably be able to address any question that Brandon doesn't have, he'll probably be able to address too. So, so without further ado, Brandon, please take it away. Oh, wait, I'm sorry, before I say that, sorry, housekeeping, I forgot housekeeping. So I think what we're going to do is we're going to run through the webinar. If you have any questions, Remy Dar, who is also on this call and is the program coordinator in the Office of Research, is going to be assisting in this. But if you have any questions, put them in the chat. You can certainly feel free to put questions in the chat as the presentation is running, and we will make every effort to get all of your questions. We've left plenty of time at the end to have questions. So any questions you have, we will go through the chat and make sure that we get to all your questions at the end. So having said all of that, Brandon, please take it away.
Good afternoon, everybody. Thank you for joining us today. I will be giving a presentation on the NIH R series, a general overview of those applications. I do want to point out before I get into all of this that I'm only going to be highlighting kind of the more, I won't say popular, but the better known R series research programs. There are still a number of other programs available, and I've included a link at the end of this presentation where you can go on to the NIH website and view all the program opportunities that they have available, which might fit your research initiatives. I also want to highlight too, many people think that NIH caters only to the biomedical sciences, but that's not actually the case. They have a number of opportunities that are appropriate for other disciplines: chemistry, biology, engineering, the behavioral sciences. So if that applies to any of you on this webinar today, definitely look further into the opportunities that are available. So we'll get started here. I've just captured a few, the R01, for example. And let me back up also. I usually don't read directly from my slides during these presentations, but given this format, I'm actually going to, in case you all aren't viewing the presentation at the same time that I'm speaking. So the first one we'll go over is the R01, and it supports discrete, specified, circumscribed projects to be performed by a named investigator in an area representing his or her specific interest and competencies. The R03 is the NIH Small Grant Program, and it supports preliminary short-term projects, and this particular program is non-renewable. The next one is the R13, the Conference Grant. So this one supports recipient-sponsored and directed international, national, or regional meetings, conferences, or workshops. The R15, also known as the NIH Academic Research Enhancement Award, supports small-scale research projects at educational institutions that provide baccalaureate or advanced degrees for a significant number of the nation's research scientists, but that have not been awarded or have not been major recipients of NIH support. The R21 is the NIH Exploratory Developmental Research Grant Award, and it supports the development of new research activities in categorical program areas. And support is generally limited in both funding and time. The R25 supports education projects, the development and implementation of a program as relates to a category in one or more areas of education, information, training, technical assistance, coordination, or evaluation. So just some things to keep in mind before applying. Become familiar with the activity code. R01, R15, R25, etc. As I said before, there is a link at the end of this presentation that has additional information. And also read carefully and fully familiarize yourself with the FOA, Funding Opportunity Announcement, PA Program Announcement, and any other supplemental guidance that has been provided. So NIH will release the FOA, but there's often notices of additional information that it could be revisions to that FOA, it could be general guideline revisions. So there's a couple of things that you need to consult when preparing your application. And I think most importantly is to always feel free to be in contact with the program or scientific officer of the awarding component at NIH to which you wish to apply. They are eager to provide insight, and they're an amazing resource during your application process. So even if you're not sure yet that you want to pursue an R01, for example, give that program officer a call and run your idea by them and just get some of their feedback and thoughts. And definitely stay in touch throughout the process if necessary. So the specific FOA instructions will always supersede the general application guidelines. And I'm sure you all realize that that's the case with most any grant proposal. Every agency has general guidelines, but the specific FOA is the one to follow the closest. And unless otherwise specified in the FOA, follow the standard instructions as well as any additional program-specific instructions. And then, as I mentioned previously, always be mindful of any significant changes or notice of NIH policy changes. And those are usually indicated, it usually says NOA and then a number correlating to the FOA that you're looking at. And that just lets you know that there have been additional guidelines or revisions to the particular FOA that you're looking at. And currently NIH is using Forms F for the application form set. If, depending on your institution, how you submit the applications, at Lehman, for example, we use a system called Cayuse. And so it updates the forms automatically. But if you don't use a system that updates automatically, just be mindful of the application you're using. And your Office of Sponsored Programs can assist with that as well.
Brandon, can I just add a couple comments on how
yeah.
I use that FOA?
Sure.
So if I have a research project and I want to get it funded, the first thing to do is actually find the right mechanism for funding it. And in the FOAs, there'll be a list of types of research programs that they will fund. So you go ahead and you look at the FOAs for different things. And, like, for example, I looked at one which was a software development grant for drug discovery. And it said basically what types of programs and what types of research agendas they wanted to fund there. And then if I decide to go for that particular program announcement or that particular FOA, I look at it, and then I also frame my research agenda, my specific aims and my research plans with regards to what they're actually looking for there. And so I keep that in the background as I'm writing the grant. And that's important because you want to make sure that your research agenda is something that fits in with the actual funding opportunity that's there.
Thanks, Tom. So as you start the preparation process, again, follow the process in your institution for notifying the Sponsored Programs Office and your intent to submit, and then determine the submission mechanism that your institution uses. So as I said before, at Lehman, we use Cayuse, but NIH has a portal called Assist, there's grants.gov and so forth. So your Office of Sponsored Programs can let you know which portal is to be used. And then the next thing you're gonna do is determine your application type. So I've highlighted the three main types. There are a few other categories that are not pertinent to this presentation, but there's the new, the renewal, and the resubmission. So new application is just that, a brand new application that you've never submitted before. The renewal application is an instance where you were awarded the first time and you're up for renewal. There's two types of renewal, competitive and non-competitive. So if, for example, you have a 5-year R01, 4-year R01, whatever the case may be, every year within that project period is considered a non-competitive renewal. At the end of the project period, you have the option for a competitive renewal. And then a resubmission is an instance where you submitted a new application, but it was declined for funding. And so you're resubmitting the application that was declined. In a resubmission, you always have to address the reviewer comments and let them know how you readjusted your proposal to speak to the concerns that they raised in the review process. And then you develop your application components. We're going to go over the main components of any NIH R series, and that's the project summary and abstract, project narrative, the specific aims, the research strategy, and the bio sketch. One thing that's really important that I try to give the faculty and PIs a heads up on is if you're doing collaborations of any kind as part of your proposal, start that work at the beginning. Get the information you need from your collaborators, letters of support, budgets, budget justifications, scopes of work, all of those things if you're going to be issuing a subcontract to these collaborators. The more you have up front, the easier this process is. Many institutions have their own internal review process for subaward agreements. And so there's a timeline that needs to be followed. So the sooner you start that, the better.
Yeah, and you need their NIH bio sketches, and often your collaborators will have those. But the bio sketch is often a place where you can add additional information about the grant proposal itself. So it's nice if your collaborators have the time to adjust their bio sketch to the aims in the research program in your grant. And so the more time that they have to do that, the better. You know, in general, because we're so stretched for time, it's often done at the last minute. But if you can do that ahead of time, that's really, I think, useful.
Absolutely. The other thing I want to point out is if your proposal requires any kind of demographic data or institutional data, begin working with those appropriate offices, whether it be institutional research or elsewhere, to begin gathering that data. At Lehman, I really try to give our IR office a 3 to 4 week lead time to provide data that we might need. So be mindful of that as well. And then start developing your budget. There are 2 types of budgets under NIH. It's a modular budget, and that's for budgets of $250,000 or less in direct cost. The next type is the detailed budget. So of course, anything in excess of $250,000 in direct cost. And I'll go over the details of that a little later in the presentation.
Can I just add one more thing about the letters of collaboration? It's nice to have it in the beginning, but you often don't. And so as you're writing your research plans, you want to be thinking about the weaknesses that you see in it. And if you were a critical observer, what would you say are the weaknesses in this actual proposal itself? And so often as I'm writing the research plans, I'll think, wow, I don't have the right experimental validation. I can't do it myself for this particular, for what I'm proposing here. I can get a letter of research that's going to sort of fill in that gap, or a letter of collaboration that'll fill in that gap that can be either funded or non-funded, where they would say, yeah, this really fits in with my research agenda, and I'd be very interested in going ahead and doing these types of things. And so it's a way of sort of filling in some gaps, and then in the actual research plan you have. And I think as you're writing your proposals, you should always be thinking about how is this going to be criticized, and trying to preempt those criticisms as best as you possibly can.
Excellent points. And then finally, just as a note on the preparation process, work closely with your sponsored programs office just to ensure compliance with attachments, internal policies, CUNY policies, all of that kind of thing. Your sponsored programs office is an excellent resource for kind of the administrative lift of these proposals. And then also be sure that you're in compliance with your institution's internal deadlines and review process. So now we're going to talk about the attachments. Arguably, one of the most important components of the application is the specific aims page. It is kind of that first page that you really should complete before anything else, but it also works in tandem with the research strategy, and Tom will talk about that in a moment. But the specific aims page is limited to 1 page, and it's kind of that point where you grab the attention of the reviewer and give a synopsis of what you're doing. And then the larger narrative is the research strategy. And so it's the technical component of the proposal where you describe your research rationale and the experiments that will be conducted to accomplish each aim. The allowable page length varies by activity code. I've included page lengths later in the proposal, but the RO1, for example, has a 12-page limit on research strategy. And then there's the biographical sketch. And it's essentially your CV in NIH format, but it is more detailed, and Tom can speak to that a little as well. But you'll indicate your education, your career path, research and publications, as well as a personal statement on your work. And it is limited to 5 pages. I do have an example of that, which I'll show a little later in the
All right, can I spend a minute or two on each of these?
Totally.
All right, so the specific aims is the first thing that most people are going to read in the review panel. It's where you have to get them excited about the project. And so the specific aims is mine are generally, it's 1 page long. Mine are generally about 2/3 of a page with the introduction and background, and which is writing, which is to try and get the reviewers excited about it. Have them realize that it's important, it's well planned out, will have an impact, it's a significant problem. You have to sort of try and get all of that into this 2/3 of a page. And it has to be in line with the program announcement or the FOA. And then the specific aims are, and then the actual specific aims are the individual elements of your research strategy. I find that mine I generally have between 3 and 5 of those, but I'm not sure what the exact guidelines are for that. The biographical sketch, one of the things that's important to put in, could you put it back up? Thanks, Brandon. One of the things that's important about the biographical sketch is that in the beginning of it, you have an opportunity to outline your contributions to science. And one of the things that you need to convince the committee of is that you're capable of doing the research and being successful in the research that you're proposing. And so the contributions to science portion can be a mechanism to communicate that to the review panel, that you are accomplished and you have the skills and background that let you do that. And with regards to the FOA, I'm just going to paste a web link in here. And we'll go back to that. So one of the things that I like to do if it's an established program announcement is I go to the project reporter on the NIH and I found out who got funded in. I'll do 2 things. One, I'll find out who got funded in from that program announcement and see whether or not they're my peers. They're people who are doing the types of research that I'm doing. That can generally be a good thing, though you don't want to entirely overlap with a proposal that's already been funded. And the other thing is I'll actually look up my peers and find out people who are doing research that's similar to what I'm doing and find out what mechanisms they've been funded under. And so sometimes I'll look up the leaders in my field and say, okay, how did so-and-so get funded? I'll look them up. You can search by their names. You can search by grant mechanisms if you follow that link in the chat. And not only that, you get to read their abstracts. So you get to see how they posed or how they framed their research program. And that can give you ideas about how to also frame your own proposal itself. That's it.
Thank you. And 3 other components that I'll touch on. The project summary and abstract. This is a succinct and accurate description of the proposed work. It should be written for a scientifically literate audience and will be made public in the event of award. So this is actually limited to 30 lines of text. The next one is the project narrative. This is a description of the work that should be written for a layperson to understand and will also be made public in the event of the award. And it's limited to only 3 sentences. And the introduction, this is only required for resubmission and revision applications. It's oftentimes in a letter format. And this document introduces the project and fully addresses the previous reviewer's concerns or justifies the need for revision of a previous award. And it's limited to 1 page.
Yeah, with the introduction, if you're resubmitting, it's a very important letter, because the review committees at the NIH are going to have the reviews from the original submission of the grant, and this is an extra page that you get to communicate that you fully addressed all the concerns of the original committee. You want to pack it full, be very polite and respectful, and all of that. Don't let, even if you're upset by the comments, and everybody's upset by the first time they get the comments, you have to make sure that you don't, none of that comes through in the in that introductory letter itself. Even if you think the comment is entirely off base, you need to communicate that you've addressed each of the major concerns that were commented on by the committee to begin with, and the supervisor of the committee.
So now we're going to speak in detail about the specific aims statements, and I'm just going to go over some of the summary and let Tom talk about his experience with this area. But it is one of the most important components of the application and should be completed before anything else. It's also what will grab the reviewer's attention and assist them in determining the feasibility of the nature and scope of your work. And it provides a roadmap for your research strategy. Again, it's limited to 1 page. And some tips for a successful specific aim statement. The introductory paragraph should capture your reader's attention, state what is currently known about the specific field, address the gap in knowledge, and outline the critical need. The second paragraph should address the what, why, and how of your project, state the long-term goal, hypothesis, and proposal objectives, rationale, and qualifications. And then the aims themselves should be clearly titled, 2 to 4 sentences each, and should be easy for the reviewers to read. As far as the number of aims that you have, it's up to your discretion. I've found on average there's 2 to 3 aims per project, but there could be more. And then the final paragraph should state what is innovative about your project, the expected outcomes, and the broader impacts. So I'll let Tom talk a little bit more.
Yeah, I mean, I like that summary. Treat it like a checklist, and go ahead, write it, see whether or not all of those conditions are met. I would just add, it should more than capture your reader's attention. It needs to get them excited about the project. Yeah. It's the part, where I have some reservations. I don't feel I'm very good at overselling my work, but it's something where in the field, if you go ahead and you're the salesman, this is the place where the salesman ability is important. And I know people who are very good at making their research sound like it's really incredible, and this is where it's done. And if you can generate that excitement at this stage, the reviewers are going to read your research plans with much more scrutiny and much more involvement, which is overall is a good thing. One of the things about this is that you want to put it in line so that's in line with the funding opportunity. So you make sure your specific aims, each of your specific aims fits within the program announcement or the fund FOA, and people say that or one of the comments Brandon and I were reading, or one of the books said this should be done first. Well, I agree it should be done first, but invariably as I'm writing up my research plans and seeing how each and really solidifying what I'll actually be doing, which is, I think, the advantage of writing research proposals is it really makes you structure your research better. As I'm writing that up and realizing, well, this is the way that it needs to be done, I will invariably go back to my specific aims and restructure them. So it's good to have it as an outline to begin with. I can't think of a single proposal that I've written where the specific aims were not revised 2 to 3 times during the writing process. And it's something that you want to think about. One last point, sorry if I'm rambling a bit, but you're going to have 3 to 5 specific aims. You want to make sure that the success, so if you have something where there's success, where there's some doubt with regards to the success of the specific aim, you don't want that to kill the other aims. So you want to try and write your aims in a way such that even if the first part of the first specific aim ends up not resulting in what you would like, that the second and third aims still have the impact and significance. And that can often be difficult. But if you go ahead and say, the first part of this is I'm going to identify this molecule, and which is something that I would do. And then once we find that molecule, I'm going to do this, this, and this to it. If you don't find that molecule, your whole thing is shot. Your whole proposal is shot. And so if you can go ahead and show that each specific aim is synergistic with the other aims, but not dependent upon them, that would be an overall good thing. It's not an easy thing always to do. That's it, Brandon.
Okay. And now on to the research strategy. Again, this is the technical component of the proposal where you describe your research rationale and the experiments that will be conducted to accomplish each aim. Made up of 3 subsections: significance, innovation, and approach. Organization within each category is at the proposer's discretion. However, it should follow a pattern similar to that of your specific aims. Use clear headings so that the reviewers can easily find what they need, and the allowable page length varies by activity code. So Tom, do you want to talk a little more about this?
Yeah, I mean, I think in general, the main part of this is the approach section, and that's gonna be divided into each of the specific aims. The significance and innovation are short. The significance, you have to say basically why the research you're doing is significant. I tend not to have a problem with that. Explaining to a committee why your work is innovative is just, I always have problems with that. I never really know what to do with it. It's like that should be in the research plans. How does this distinguish you from other work? I generally feel very uncomfortable writing that section. So I don't have much to offer there. I don't think. It needs to be clearly communicated. I don't think, the research strategy should be something that, the people are expert in the field, but they're not you. And so they don't know all the details. So they really need to be able to follow what you're going to do and understand why each part of your approach is significant and important for the problem. I think that's it, Brandon.
Okay, thank you. And here is just a snapshot of the page limits. Again, this link is in the proposal or in the presentation for you all to look at later. I apologize if the font is a little small. I enlarged it as best I could.
I'll just add here that I always do the project summary and / abstract and narrative as the very last things. As soon as everything else is finished, I'll go ahead and write those, because then you know exactly what you're writing about. And any unresolved issues by that point are resolved.
Excellent point. And just a few housekeeping kind of things to keep in mind. All of the attachments must be submitted in accordance with the FOA or the program announcement. The documents must be sized at 8.5 by 11. Documents that are incorrectly sized will be rejected by ERA Commons. One area that I encourage faculty to pay attention to are the letters of support. Oftentimes, if they're scanned in as a PDF, if your settings on your scanner or in your PDF program are not exactly as they should be, oftentimes the letters are either too large or too small. And I'm talking very minimal differences. It may be such that it's 8.4 by 11.38. And although that may seem insignificant, NIH ERA Commons will kick it out and reject the submission.
This is a huge pain in the ass when you have international collaborators, because they don't use
Totally.
8.5 by 11. They use A11, and I, there are online PDF resizing places, which I've spent the very last minute on a deadline way too much time on going ahead and taking the international collaboration letters and resizing them from whatever 11 to 8.5 by 11, and it's a huge pain. If possible, you can ask them to go ahead and do it on 8.5 by 11, but when it's a senior person abroad, that's sometimes a difficult request.
And I will say that this kind of thing is probably what the Office of Sponsored Programs spends the most time doing before a submission, is checking page sizes and resizing documents. It's incredibly tedious. The margins must be at least 1/2 inch on all pages. Fonts must be at least 11 points or larger, and there must be no more than 6 lines of text per vertical inch. Again, these are areas that ERA Commons will kick the proposal back. And digital images should be approximately 1200 by 1500 pixels using 256 colors, and must be readable as printed on an 8.5 by 11 page at normal scale. You must use either a JPEG or a PNG. Tom, do you want to say anything about the importance of images in the research strategy?
I mean, I use images, and we had a brief discussion about this yesterday. I used images when they communicate what I'm trying to communicate better. That's it. The documents should look nice, but basically that's what I do. Put some effort into the images. They shouldn't look sloppy, and they should be clear and understandable, and communicate what you want to communicate. And often they can do that a lot better than written text. I think that's it.
Okay.
Yeah.
And regarding the budget, again, going back to what we outlined earlier, modular budgets are required for direct cost request of $250,000 or below, and must be created in $25,000 modules. Detailed budgets required for direct cost requests in excess of $250,000 or as required by a particular FOA. One thing I will point out, and the reason I'm not spending a lot of time on budgets, is, for example, the R01 is not really capped at a budget. They ask you to propose a budget that is reasonable for the work that you're accomplishing. And so in that respect, you may have a request of $250,000. You may have a request of $450,000. And the good thing about the modular budget is it's just kind of a summary request to the NIH. It's not a line item budget. The line item budget would be created internally for, the Office of Sponsored Programs or the Research Foundation to help administer the program, but it's nothing that you have to submit to the NIH. Detailed budgets, on the other hand, are the line item budgets. And so, they are much more time consuming and detailed than the modular budget. Oh, go ahead.
No, go on. I'll do it when you're done.
Okay. I was just going to say the FOA will state what costs are allowable and which ones are not. NIH also offers additional guidelines on standard allowable cost. And you'll notice the allowable costs are usually consistent with other federal agencies and their allowable cost. And indirect cost in most cases are provided in addition to the direct cost request and will be based upon your individual institution's federally negotiated indirect cost rate.
Yeah, well, I don't exactly understand it right now, but I think if somebody has a subcontract on your grant proposal, the indirect costs their indirect costs come out of of the direct costs for the grant. So that's often a reason to go non-modular. The advice I got on this from people who are much more successful at writing grants than I am, who've gotten many grants, was ask for what you need. Go ahead and make a budget and ask for what you need. I think that, or my understanding is that, the grants are based on the merit of the research. The reviews are based on the merit of the research, not by how much you're asking. If you're asking for way more than ever could be feasibly required to do the research, they're going to reject it. And you're not going to do anybody any favors by saying you can do this it's not a bidding process where you say I can do this for 200, when you really need 250. You ask for what you need to do the job right, and don't be concerned with the rest of it. And that's been my strategy throughout writing grant proposals, and it's been fine. And I've never seen, I personally haven't ever gotten, I've done one modular and one non-modular R1 that have been funded. The second one was because we had a large subcontract and we needed the extra direct cost. In every case, they've said that the request was reasonable. Keep in mind, though, while you're doing this, is that it is very standard in today's funding market that these grants are going to be cut between 20 and 25% off the board. If you ask for 5 years on an R1, there's no way you're going to get 5 years. We had a Nobel Prize laureate whose name is escaping me right now. He got a perfect score on all the reviews, which is 10s by across the board, first percentile. Had won the Nobel Prize, was working on continuations of his Nobel Prize research, and asked for 5 years and got it cut to 4 years. And so if he's not going to get it with those reviews and that background, you're not going to get it either. So you're going to get, you can ask for 5 years, but they're going to cut it to 4. I thought in one of my things, we might as well ask for 5, and then they think, well, we cut it to 4, maybe we won't cut as much budget out of it. That didn't work. And so, but generally there'll also be a cut to the direct costs that you're getting as well. And that will be between 20 and 25% of the R1. And I think that's pretty much standard in today's funding. And so, keep that in mind as you're going ahead and budgeting for the whole thing. One really nice thing at CUNY is our graduate students are very inexpensive to fund in comparison to other universities which they are funding. Our salaries are lower than theirs. So when you're thinking, wow, I'm asking for a lot here, when you look at it in comparison to what other universities are asking for the comparable number of students and postdocs, it's really a lot less. And so, I don't think they pay too much attention to the budget, but in general, the cost of doing research at CUNY is less than the cost of doing it elsewhere.
Excellent points. Absolutely. Okay, so you've submitted your proposal. Now what? Upon submission, you will receive a notice from NIH ERA Commons that will alert you to the status of your submission, whether it was accepted or rejected, and it'll give you the opportunity to verify the proposal up to the submission deadline. The verification period does not extend the submission deadline, so keep that in mind. But the verification period is just a time frame in which you can go back and make some basic revisions to the proposal. Maybe you put in the wrong image in your research strategy, or maybe you wanted to add an additional clarification in your specific aims. This period of time is when you do that. The review process will follow the timelines outlined in the deadlines and award information page on the NIH website. Again, I have a link to that later in the proposal or in the presentation. And if the proposal scores in a range to be considered for funding, a just-in-time request will be received, and you will be asked to provide additional supplementary information. This could be something like your current and pending support documents, any kind of clarification that the review panel needs to make a final funding decision. Do keep in mind that a just-in-time request is not an award notice. you may receive a just-in-time request and still not have your proposal funded. If you receive the JIT, in all likelihood, it will be funded, but it is not a guarantee. So just keep that in mind. And then if it is,
you should be celebrating if you get the just-in-time
yeah,
request.
totally.
They just want to make you wait and be anxious about it for a few more months and suffer. But generally you get it. Though they won't commit to it. They won't commit to it yet.
They won't commit. I've only seen, I think, 2 instances in which someone wasn't awarded after getting a just-in-time.
Yeah.
And if you are awarded, you'll receive an official notice of award, and that's usually sent directly to the Research Foundation, but it often has copy to the PI. And here are just the various references and resources that I mentioned. One thing I do want to show you is the bio sketch. And so I'm gonna close this out for a moment.
Can I just add, I know that we're at a time limit right now. Don't be afraid of your program officer. You talk to them, they end up being nice and reasonable people, and they're excited about funding exciting science. That's a perk of their job. So you can talk to them and get advice from them. And often after your grant is reviewed, talk to them, because they're in charge of the review section, and they know what the discussion was about. And so there's limitations on what they can and can't tell you, but generally talking with them is informative.
Absolutely. Bear with me, my computer's running a little slow. Trying to pull up the bio sketch here.
And as Tom mentioned, it is 12:47, but with it, we have left enough time for questions. So please, if anyone has any questions, just type them in the chat.
Okay, and this is just a bio sketch and what the current format looks like. And this was pulled directly from the NIH website. But you'll have your basic summary up at the top, your personal statement, any positions and honors, your contribution to science, any of your published works, and any of your current support. Tom, do you want to say anything additional about the bio sketch?
Yeah, I have one that I have my standard personal statement and research contributions on, and that's nice, and you think you have it prepared in the beginning, so it's one thing you can check off. But if you do have time, it is worthwhile going in and catering your personal statement to the research proposal, structuring it so that it communicates things that you would like to have communicated in the research plans or the specific aims or the abstract and summary or everything else that you couldn't. I've put timelines in the bio sketch. I don't know if that's allowed, but generally you don't fill up 5 pages of your biographical sketch. So there is room to add additional elements here. That address I think what's most appropriate here is address your background, the fact that you can do the research and that you have the expertise to do the research, and a track record if you've been around for a while. That's it.
And I will say with the bio sketch, this is another area that if you exceed the page limit, ERA Commons will kick it back.
Yeah. And one last thing, which is a pain here. If you look at this one, it says approved through 2/28/2023. I have been dinged because a collaborator of mine sent in a bio sketch that was approved through 2018. They didn't reject it outright, thank God, but they did say this, they did mention it in the actual reviews themselves. So, and the thing is they'll go ahead. This is good. This is good for a long time, luckily, but they expire. And the next time you write a grant, it looks identical to this. In fact, you can't even find any differences whatsoever, but you want to make sure you have, the right one, because they will they do change them occasionally, and they might catch it. And they're sticklers a lot of the time. And I have heard of grants. In fact, I know of people who've had their grants rejected because they used the wrong font size. I recently saw on Twitter that some guy had his grant rejected because he put in a hyperlink in the grant proposal, and the NIH guideline says you can track hits on the web from hyperlinks, and so they don't allow them, or they have very strict guidelines for them. So be a little careful with hyperlinks if you do want to put it in, like to your GitHub or to maybe to your web pages. I don't, that have your records. Be careful. I think you can probably go ahead and link to the NIH.
So we don't really have any questions in the chat right now. There is one inquiry about us doing a webinar on the SCORE grant, and we can certainly talk about that. John Spogus was on the call earlier. I noticed he was in the webinar earlier. He's the director of the Research Foundation Pre-Award Support Office, as I had mentioned earlier. And about a year ago, he did run a day-long webinar that our office also participated in on SCORE grants. And I'm not sure, John, was that recorded? We could certainly do something again in a virtual format. I'm just wondering if the resources you had from that event are available somewhere.
Effie, I'll check. I think it was recorded.
Okay.
I believe it was recorded, and we can put it on our website and share it with everybody. It was actually quite a good seminar, because it included administrators, it included successful SCORE applicants, and so I think we can put that one up. May not be a bad idea.
Okay. Yeah, it was also good too because it broke up into groups, because there's 3 types of scores. There's the 1, 2, and 3, and it broke it up depending on which type of score you were applying for. So that was very useful too. So there was a question that came up. Yes, go ahead.
I just wanted to say that when you look at all NIH grants, CUNY is the most successful in the SCORE grants, more successful than R01s, K-series grants. Because SCORE grants are designed to build the research capacity of minority serving institutions. So we were very, very successful in SCORE grants.
There's some couple questions coming in. So there was a question about how this differs from the PSC CUNY grant. So the PSC CUNY grant is actually an internal grant program that is actually administered by the Research Foundation, and it's all internal funds, and it's all for all faculty all across CUNY and all disciplines. So this is actually this workshop is focusing specifically on National Institutes of Health, which is a federal agency funding. So is there an NIH?
Effie,
I'm sorry, go ahead, yes.
Can I add one thing about the
Sure.
about the SCORE grants? I've gotten 2 SCORE grants and 2 R grants, and the research plans, the specific aims for all of those were exactly the same, except for the SC3s, you're limited to 6 pages for the research plans. What the significant difference between them is that the SCORE grants, as John mentioned, are developmental grants. And so in the bio sketch for the SCORE grants, I add a section which talks about building the research infrastructure and the PI development plan inside the bio sketch itself. So I have a PI development for research. I have something building the physical and computational infrastructure in my lab, personal skill set development, cultivation of collaborations. These types of things resonate well because that's in the FOA. That's part of what they want to fund. They want to actually fund the science, and that's first and foremost. But the biggest difference for me was putting in for doing the SCORE grants as opposed to the R grants was putting in a development plan, putting in what your goals are for it, which by the way for the SCORE grants is to prepare you to apply for R grants.
Yeah.
And so putting in that that you're building the infrastructure and getting the experience and the publication record that's necessary to do what they want, which is to prepare you to get our funding. And that can all go in the bio sketch.
We had a question here about, is there an NIH analog to the CUNY NSF for Career Boot Camp? I believe the NSF Career Boot Camp is currently being run by Linda Vigdor out of the ASRC, but I do know that John, both our office and John's office in the past have offered similar boot camps. There's not currently an NIH boot camp. One of our former vice chancellors, Mark, why am I forgetting his last name? But he actually did a boot camp on writing a specific aim statement, which was actually
Mark Halber.
Mark Halber.
Yeah.
He ran a boot camp running specific aim statements, which was very successful, where a group of faculty got together and actually critiqued each other's specific aim statements, which actually worked out very well. But in response to your questions, no, there is not an NIH boot camp right now, but if that is something that people think would be useful, I mean, that's certainly something that we could try to pull together. This is a question perhaps for Tom or Brandon about, can you elaborate more about contractors versus collaborators? What about multi-PI grants? Can you answer that? That actually might be a question for Brandon.
So contractors and collaborators, essentially, is it a sub-award or is it not? It involves money. Are you collaborating with someone that you're giving money to? So will they be receiving money for their effort? Will they be adding grad students to their lab? Anything that you know, NIH will be paying for through the proposal would be a contractor / subcontract. Collaborators are anybody you collaborate with. It doesn't have to be, it can be paid or unpaid. Essentially, you know, someone can provide some level of support to your project and not ask for any kind of funding to provide that support. And so that's simply what a collaborator is.
And a collaborator just has a letter of collaboration.
Right.
A contractor who's also a collaborator, which is the reason for this confusion, is needs an NIH bio sketch, often needs I think a letter of support for the grant itself. They need something that expresses their involvement. I can't remember what the document's called. I don't know if that's after funding or before funding, actually. I can't remember right now. But there's an additional number of documents that you need if you're actually funding somebody who's outside of your college, Brandon, or outside
institution,
of
outside of
institution.
your institution.
Yeah, and I think, is your institution your college or is it CUNY?
So CUNY handles it a little differently than what I've been used to. I found CUNY colleges don't necessarily require the paperwork agreements between the institutions. However, when I was at Vanderbilt, we did have letters of collaboration that were signed by institutional officials acknowledging the collaboration of the project. Right. And so
often that's after that's been funded. So you need that because that's a legal contract for the finances of the grant.
I encourage gathering all of that stuff at proposal time. That way the collaborating institution is at least aware, but NIH doesn't need anything at the proposal time usually.
Yeah, and this is something that definitely your sponsored research office can help you with, the grants officers on your campus. So the next question is, what does SCORE mean and how do we access information about it? And SCORE it stands for Support of Competitive Research. I mean, and Tom and actually Brandon, you can probably address this a little more directly, but it's the program that generally you apply for before you apply for the R01, but maybe you could be a little bit more specific about it, Tom or Brandon.
Tom, do you want to take that?
Okay, so there's three separate, what are they, parts of the SCORE grant. They're SC1s, SC2s, and SC3s, and the order that you actually apply for them is very different. It's generally the SC2 is a mentored grant. It's $100,000 per year for 3 years, so it's $300,000 total. But you need a mentor for that, an established mentor for it. So you write that with somebody else. I haven't written that, so I don't know all the workings of that. Once you get that, but you don't need it, I skipped it. I went straight for an SC3, and that's $75,000 a year for 4 years. And that's a 6-page plan. And one thing that John mentioned is we're much better at getting R1s than we're much better at getting score grants than R grants. That's absolutely true. They're much less competitive. We are not, when you write a score grant, your competition are other faculty at minority serving institutions or other institutions that qualify for the SCORE program. You're not competing with MIT. You're not competing with Harvard. You're not competing with UC San Diego and all the top flight R1 institutions in the country. And so the funding rates for SCORE go all the way down something of the level of 30%, whereas an R1 nowadays is about, depending on which area you go into, is around 14%. 8% to 15 to 20% funding for the R1 grants.
I just wanted to point out it's 1:02, and I just want to thank, I know some people are leaving.
Okay.
We can certainly, both Brandon and Tom said they had a few more minutes to answer some questions after 1 o'clock, but for those of you that need to leave, thank you so much for joining, and we will, as I had mentioned earlier, both the presentation and the webinar will be available on our website. So thank you so much. I'm sorry to interrupt you, Tom. You can complete that thought.
Yeah. Well, the last one is the SC1. Which is at the funding level of an R1. From my experience, and I started at Lehman College 10 years ago, I was not ready when I entered the university to start at that funding level. I didn't have the infrastructure in my lab. I didn't have graduate students. I didn't have the things that actually I think would lead to success to get to start with an SC1. I didn't have real startup funds, or not a significant amount of startup funds to actually start a research program. So I think, the guidelines of going through the SC2 and SC3, building the infrastructure, building up the program that you would want, is one that really fits in well with the CUNY system. I just saw a comment that says you need to go through an internal competition to select the applicants for submission. One problem that we have at the CUNY campuses is that these are very popular. They're a lot easier to get. They're thank God for them. For me, it really made the difference in building a successful research program for me. At Lehman College, we haven't hit the maximum number of grants that you can have at a given college, which I think is 20. I know that at Brooklyn, you have to go through a competitive internal review in order to get on it, or you get on a waiting list. And not only that, you have to apply for the SC1s because they're $250,000 direct costs with the corresponding 65% graft that CUNY will take, or sorry, the 65% cut that goes to overhead and supporting research. And so, that's close to $150,000 a year in overhead, whereas the overhead for the SC3 is $42,000. But the limit is 20 total. So at Brooklyn, I think they don't allow you to write SC3s. And I'm not at Brooklyn College, but that's what I've heard, secondhand. So, some of those aren't available. Lehman's competitive in that way because we can apply for any one of them because we haven't hit that limit. But I'm sure that when we hit that limit, we'll have to have an internal competition. So I think Brooklyn and Hunter, and I would guess City College would be ones where you would have to go ahead and see if you can get them at your university. I don't know if any other school has hit.
Yeah, those
limit.
schools all has, as the commenter mentioned they have an internal process for, selecting who can apply for the score. So it looks like there's a lot of support for an NIH bootcamp. It sounds like maybe we need to do a webinar on, and I'll talk with John about this, and even Linda Vigdor, who runs the NSF Career Bootcamp. Maybe we'll try to do both an NIH, maybe an R01 bootcamp, and then also a program about SCORE grants, which would seem very useful. We just have one question. I know this came up before, but somebody asked a question: of these grants limited to biomedical research? And Brandon, I know that you have a response for that question.
So no, they're not. Many people think they are, but NIH has a very broad range of disciplines that they will fund. And so when I was at Vanderbilt University, I was in the School of Engineering, and we had a huge portfolio of NIH grants. And so, if you think your discipline fits, by all means, look for a mechanism to fund through NIH.
All right. Well, great.
Can I just add one more thing? I think, Brandon, you have the link to the book that I sent you.
I do.
In the final page of the presentation. But there are books. The first time I wrote a SCORE grant, I was actually at San Jose State, and one of my reviews was, read a book on how to write a grant. At first I was angry and upset, but in the end I realized, wow, that was pretty good advice, because I had really no idea what I was doing. And I got a book. I gave a talk at Albert Einstein when I came to Lehman College, and they had somebody come in and teach from the NIH, teach from this book. And I read that book, and I get in Brandon, you have it in your grants. I
just put the link in the chat.
Oh, there it is. And I thought it was fantastic. And you read through it, I read through it every single time I write a grant proposal, and I reread it and think, these are the important elements that I have in each section. And you have a checklist, and you just check them off. Have I shown it's significant, in this part? And that really helps structure the entire proposal.
Well, fantastic. That's really useful. Well, great. Well, thank you so much, Brandon and Tom. It is now 1:08. We really appreciate your insight and your time and your expertise. And we'll see you all next month at the next Monday, Friday.
Good luck, everyone.
Thank you. And please,
Thank
if you have any questions, do not hesitate to contact me or anyone on this call. Thank you.